Hi students, in the last class we discussed about separation of plant pigments from the leaf extract, right? In that case we identified there is xanthophytes are there, carotenoids are there, and chlorophyll B and chlorophyll A, right? That is about the separation of plant pigments, right, by the paper chromatography technique, right, by the usage of mobile phase and stationary phase. We did the pigment separation process, right? So that one is the separation of plant pigments. Coming to the next one is the photosynthetic pigments. Coming to the next one is the photosynthetic pigments. See, both are same, but we are giving the name here. The photosynthetic pigments means which are involving photosynthesis process. See, these pigments can be classified into two groups. The total pigments can be classified into two groups. One is the reaction center, where the final reaction takes place. That particular location is called as reaction center. Other one is called as, okay, the remaining part of the pigments are called as accessory pigments. So totally I said here the two groups are there. One of the pigment is called as reaction center, remaining contents are called as accessory pigments. So always within the reaction center there is a chlorophyll A pigment is present, no doubt about it here. Always the reaction center chlorophyll A is present. See, I like to say photosystem 1 is there and photosystem 2 is there. So photosystem 1, the reaction center is the chlorophyll A. Yeah, photosystem 2, the reaction center is the chlorophyll A. Both the cases, in any case, the reaction center means that is one of the specialized chlorophyll A molecule. That is one of the specialized chlorophyll A molecule. So here we are discussing about the chlorophyll A. See, this particular pigment is called as reaction center. This particular pigment is called as reaction center. One is called as the reaction center. So then all of you keep the heading also here, reaction center. So reaction center, that is nothing but chlorophyll A. Okay, see here, universal pigment. This is the universal pigment. Whatever the plants are there, in all the plants contains the major pigment is the chlorophyll A. That's the reason why this one is called as universal pigment. And this is one of the chief pigment, means this one is always present, and this one is abundantly present. This one is abundantly present. And next one is that blue-green color. See, we discussed there is a chlorophyll B is there, green color complex, and there is a chlorophyll A is present here. This one is that blue-green. So somewhat light color differentiation is there. So in the first year also we discussed there is a chlorophyll A, chlorophyll B, and chlorophyll C, and chlorophyll D is there. Then what is the major difference between these chlorophyll molecules? So all these are the four chlorophyll molecules, no doubt about it. So the center one is that magnesium. The center one is the magnesium. So here this one is the one. There is in ferrule rings are there, 1, 2, and 3, and next one is the 4, right? And these are the ferrule rings which are connected one on one like this. Right? This is the central atom. This is the central atom. The central atom is the magnesium ions. And all of you remember there is a phyta side chain is there. There is a phyta side chain is there. See, whatever the components are, whatever the chlorophyll molecules are there, in all the cases, majorly, whatever that the main structure is present. This is the main structure. This one is called as head part. So the head part is similar. In all the cases, mostly the head part is the similar. Right? There is a tail part is there. The tail part is the quite different. This one is called as phytal side chain part. The phytal side chain part is quite different from the remaining all components. Means here, what I am telling here, there is a chlorophyll A having one kind of phytal side chain, and chlorophyll B having another phytal side chain, chlorophyll C another, and chlorophyll D another. So here, whatever the chlorophyll pigments are there, all these chlorophyll pigments contains that head, and there is a phytal side chain parts are there. In all the cases, the phytal side chains are definitely different, right? So that is about that different types of chlorophyll molecules, different types of chlorophyll molecules. So the color is the blue-green color, right? And coming to the next one is the accessory pigments. See, we discussed already regarding to that accessory pigments means we know, yeah, there is in TV, right? TV channels are there. We know about the TV antenna. We know about the TV antenna. See, there is a receiver is there. Within that TV antenna, there is a receiver is there, right? This is the receiver point. This is the receiver point, right? So, along with that receiver, there is a long, round disc-like structure is present. Long, round disc-like structure is present. So, what is the use of this disc-like structure? See, the receiver is chlorophyll A. Here, the receiver is the chlorophyll A. The disc-like, the disc-like structure is nothing but the accessory pigments. The disc-like structure is nothing but the accessory pigments. So, whatever that accessory pigments are there, the accessory pigments initially absorbs the light energy. That light energy is subjected to the reaction center. The light energy is subjected to the reaction center. So the concept is very easy here. Whatever that the accessory pigments which are covers around the reaction center, right? At the center point, reaction center is present. Reaction center is covered by the accessory pigments. That accessory pigments majorly forms the shield-like structure, right? Which are supporting for the absorption of light energy. Okay. Then once the light energy is absorbed by the accessory pigments, that light energy is supplies to the reaction center. So there is an accessory pigments are there. Here there is a different types of accessory pigments are present. Phycobilins are there, right? Chlorophyll B. Here chlorophyll B is here accessory pigment because reaction center is only always chlorophyll A, right? So here carotenoids are there and xanthophylls are there. All these are the accessory pigments. So here the phycobilins are there found in algae. Phycobilins are found in algae. There is a phycocyanin. Within that phycobilins, there is a different types are there. Not much more here. There is 3 types are there. Okay, these are especially found in algae. I thought we already discussed regarding to these pigments also. There is a phycocyanin is there, blue color complex. We know about that the cyanin. See, actually the cyanin indicates here the color complex is blue. The color complex is the blue, right? Allophycocyanin. See, the phycocyanin and allophycocyanin which are especially forms the blue color complex, right? And phycoerythrin, that one is seen in red algae. The color is the red color. So based on the type of the pigment, the plant pots exhibit different types of the color. So some are exhibit the blue color and some are exhibit the red color. If the pigment is varied, especially the phycobilin pigments are varied, the color also varied. So which one? Okay, in certain cases both these are present. Which one is the dominated? That color is mostly exhibited, right? So that is about that phycobilins. And next one is the chlorophyll B. Here the chlorophyll B is the green color complex. Chlorophyll A is a blue-green color complex, right? So quite very little variation is there. And carotenoids are there. The carotenoids, we know about the carotenoids, right? Carrot color is the orange color because of the beta carotene, right? Here there is an orange color complex is formed. Xanthophylls, which forms the yellow color complex. So here the very we know about all these pigments. All these pigments are majorly forms the shield-like structure around the reaction center, which are supporting for the absorption of light energy, followed by certain type of production also given by the accessory pigments, right? So we have to discuss about the functions of the accessory pigments. We already discussed, right? Here absorbs light energy and transfers to the chlorophyll A. See, whatever that accessory pigments are there, the accessory pigments absorbs the light energy. That light energy is transferred to chlorophyll A pigment. Because chlorophyll A pigment is the reaction center. This is the reaction center, right? And protects the chlorophyll A. See, I said the reaction center is protected by the accessory pigments which forms the shield-like structure. Okay, especially from the photo-oxidation. When these pigments are exposed to the high light intensity, in that case, the pigments are damaged. Presence of high light intensity, okay, the pigments are damaged. That's the reason why this one is called as photo-oxidation. This particular process is called as photo-oxidation, right? So this one is also called photo-bleaching. The process is also called photo-bleaching. Yeah, this one is also called photo-bleaching, right? See, especially this step is called as photo-bleaching. Okay, this one is also called photo-oxidation, right? So that is about that accessory pigments, right? And these are the accessory pigments and functions of accessory pigments. I already said one is the absorption of light energy, other one is the production. Right? That is about the pigments. Yeah, the next one we'll discuss about the next topic. Right? Coming to the next topic is the pigment activity. See, pigment activity is nothing but we know about the light and we know about the pigments, right? Pigments are responsible for the absorption of the light. See, within the light, if the light is subject to the spectrum, right, the light will be divided. We know the light will be divided into vector, means within the light different types of colors are there. So within different types of the lights, within different types of absorptions are exhibited by the pigments. Simple concept. Within the light, different types of colors of the lights are there, or different colors are present. See, one color light is absorbed by the pigment in one way. One way means here. During photosynthesis process, high rate of photosynthesis is seen in blue color light, right? In the same manner, okay, some other color, okay, there is a low color photosynthesis will be observed. See, high rate of absorption seen within the blue color region, right? So low rate of absorption, some other color. So why I am specifying here, the pigments also shows the activity in different types of lights. Different types of lights means within the light energy, within the light source, again different types of colors are there. So within different types of colors, different types of absorptions are exhibited by the plant pigments. So that's the reason why this heading is given as pigment activity. The pigment activity is nothing but absorption of light. Pigment involved in the absorption of light. So the rate of absorption is different from one type of light to another type of light. So now we are discussing about that one. See, here we are estimating by the spectrophotometer. That is the one of the device. Okay, by the usage of that device, we get different types of graphs. See, one of the chlorophyll A pigment we are taken. Chlorophyll A, one of the pigment we are taken. Right? The pigment is subjected to the absorption spectra. So finally we get one graph. So the chlorophyll B or chlorophyll A or carotenoids shows one particular graph, which produce one particular graph. So at 400 nanometers, one type of absorption. At 500 nanometer, another type of absorption. At 600 nanometers, another type of absorption. So here the type of based on the type of the light, the rate of absorption is altered. Based on the type of the light, the rate of absorption is altered. So here, graph shows the ability to absorption of light by the pigments at different wavelengths. See, we know there is in 400 to 700 nanometers. The 400 to 700 nanometers, that is the light is only utilized by the plants, right? See, this is the Webster range. This range is the Webster range, right? See, this one is also called pair. This one is also called pair. Pair is nothing but photosynthesis active region. Photosynthesis active region. So within this region only okay, photosynthesis will be takes place. There is in IR radiation is there, UV radiation is there, and different types of radiations are there. All that radiations are not utilized for the photosynthesis process, but only a pair reason the photosynthesis will be takes place. So here there is an graph is there. For suppose, okay, for this graph, this is one of the different wavelengths. This is the different types of wavelengths, right? So this is an absorption. See, there is an x-axis is there, there is an y-axis is there. Upon that x-axis, I mentioned here different types of wavelengths, right? Light wavelengths, right? Here on the y-axis for this blue color graph, absorption, rate of absorption, right? So here we are constructing the graph, okay, what versus what here, wavelengths, okay, wavelength, light wavelength versus absorption, right? So between these two we are constructing the graph. X-axis we are taken one points and y-axis taken another points. So if we construct the graph, say at particularly near to the 400 region, right, one of the maximum absorption is seen. One of the maximum absorption we can observe. This is the maximum point of the absorption. This is the maximum point of the absorption, right? After that again around near to the 500, okay, after the 450, again the rate of absorption is decreased up to, okay, 650 or 680, like that the rate of absorption is low. Within that region the rate of absorption is low. Again absorption, the rate of absorption is raised, okay, near to the 700, near to the 700. After the 700 again the rate of absorption will be decreased. After the 700 the rate of absorption will be decreased. So here what I am telling, especially the maximum absorption seen around close to the 400 nanometers, right? See, the other one is that again 700 nanometers there is a maximum absorption is there, but in between these two the maximum absorption at 400 nanometers only. The maximum rate of absorption at the 400 nanometers only. So remaining are, okay, remaining also shows the graph, but that points are not forms any high peaks for the absorption of light energy. So that is about that absorption process, absorption of light energy by all the types of the pigments. What are the chlorophyll A, chlorophyll B? So this is the common graph line. This is the common graph line for all the pigments. So other one is, this is the different types of wavelengths. This is the absorption and this is the rate of photosynthesis. This is the rate of photosynthesis. The red line indicates here rate of photosynthesis. Simple. More light absorption is there, more amount of photosynthesis will be takes place. Less amount of light is absorbed, less rate of photosynthesis is exhibited. So here whatever that the red line is there, so the red line automatically follows that blue line, because see, this is the top point, this is the top point, so maximum rate of absorption, and slowly the rate of photosynthesis is decreased. Again the rate of photosynthesis is increased, because where the light absorption is more, there photosynthesis rate also high. The rate of photosynthesis also high. So that is about that rate of photosynthesis process. And commonly all of you remember high light absorption, high rate of photosynthesis, and oxygen evolution also more. Where the more amount of photosynthesis takes place, there definitely more amount of oxygen evolution takes place. More amount of oxygen evolution takes place. So that is one of the common point, right? So that is about that the first graph. And if we observe the three types of the pigments, there is in chlorophyll A and chlorophyll B and carotenoids are there. So we know about the chlorophyll, that is one of the blue-green color complex, right? This is one of the blue-green color complex, and another one is that there is a green color complex, right? And this one is also called yellow-green also. The chlorophyll B color also called yellow-green color. We know about the carotenoids. That carotenoids color is the, right, orange color. The carotenoids color is the orange color, right? And if we observe, okay, if we observe the rate of photosynthesis, rate of light absorption process, if we observe the rate of light absorption process, so here the carotenoids are very, very less absorbed to pigments. And see, we know about this one is the chlorophyll B is greater than chlorophyll A is greater than carotenoids. So in all these pigments, the maximum absorption is exhibited by the chlorophyll B. This one shows the maximum absorption. So maximum absorption shown by the chlorophyll B pigment, right? And the least pigment is here carotenoids. The least absorbing pigment is the carotenoid pigments, right? So that is about the different types of pigments. Within the pigments, if we compare the rate of absorption, more amount of light is absorbed by the chlorophyll B. And other one is the rate of photosynthesis process. And in this case, we know we already discussed where the more amount of light absorption takes place. Where the more amount of light absorption takes place, there definitely high amount of photosynthesis takes place. All of you remember, this one is also compared with the oxygen evolving process. For suppose here this is the x-axis, right? And this is the y-axis. So here we are just comparing. Okay, we just forget about the photosynthesis process. And this is the graph. Okay, these are the different types of wavelengths, and this is about oxygen evolving process. So based on oxygen evolution, we can explain the rate of photosynthesis also. Okay, at which wavelengths more amount of oxygen is evolved, at that particular point also more amount of photosynthesis will be takes place. So the simple explanation is, if we take and just this graph is completely related to the oxygen evolution process, and this one is related to that different wavelengths, and this is the oxygen evolving indicator on the y-axis, right? So here this is the graph. So where the maximum rate of light is absorbed, there high rate of photosynthesis will be takes place. There high amount of oxygen is evolved. So this is the graph. So we already discussed very close to the 400, and very close to that 700. These 2 points are the maximum points. In between these two, 400 point is the maximum point for the more amount of photosynthesis rate and more amount of oxygen evolution process also, right? So that is about that the photosynthesis, oxygen evolution, and absorption pigments, absorbing pigments, right? So that is about the pigment activities. So this is one of the big, so in all these pigments, which one shows that maximum absorption? Chlorophyll B pigment shows the maximum absorption, right? So that is about the pigment activities. In the next class, we'll discuss about that the components of the PS1 and the components of the PS2 and accessory pigments. In the next class, we'll discuss it.